Let's get back to the session. We have another change from the old program to the new program. We're going to have Rafael Gomide from Enterprise to tell us about the company here. So please.
Você aponta para lá. Entendeu? O laser vai lá, né? Mas para passar você tem que apontar para lá.
Tem que apontar para lá.
Lá atrás.
Então pode começar. Hello. Good morning, everyone. Well, first, I'd like to thank for the opportunity to be here today. Unfortunately, I couldn't watch many of the talks that we had, but the few ones that I did were simply great. So it's truly an honor to be here sharing this stage with many of you here. Second, I'd like to briefly introduce myself. My name is Gabriel Gomide. I am a bioinformatics specialist at Enterprise. Enterprise is a company with over 30 years of experience in commercial representation, specifically about scientific product brands. And one of these brands is the Oxford Nanopore Technologies, which I represent here today. I do apologize in advance because my presentation will be in Portuguese, especially to those non-Portuguese speakers here today, but please feel free to come out and talk to me later. I'll be right here outside in the hallway, and I'll be happy to help, happy to explain you about the technology. Então, bom dia. O título da minha apresentação vai ser como a gente consegue usar a tecnologia da Oxford Nanopore para a vigilância genômica e como ela é uma ferramenta muito valiosa dentro desse ponto de vista. Antes de começar a falar, de fato, sobre a apresentação, eu gosto de relembrar o tema principal, o lema da tecnologia. A tecnologia é pautada em cima de 3 pilares, que são o sequenciamento de qualquer coisa, por qualquer pessoa e em qualquer lugar. São esses 3 pilares que eu gostaria que vocês mantivessem em mente ao longo de toda a apresentação, para que no final a gente entendesse qual é de fato esse papel significativo dela na rede de vigilância genômica, de patógeno e epidemiológica. Quando a gente fala de sequenciamento, na verdade, sequenciamento é um termo mais abrangente para todo um grupo de processos que acontece. Então, começa ali desde o desenho experimental, do preparo da sua amostra, a escolha de kit, a extração, passa de fato pelo sequenciamento e finaliza na etapa de análise de dados. Aqui eu vou querer trazer para vocês um pouquinho de cada uma dessas etapas, só para a gente entender como funciona o contexto como um todo, e depois finalizar com algumas aplicações de como, de fato, a gente vê essa relevância e a importância dela em questão. Inicialmente, sobre o preparo, então, a primeira etapa seria de desenho experimental, e quando a gente pensa em processamento da amostra, logo vem à cabeça a escolha do kit que a gente precisa ter. Eu não vou entrar em detalhes nessa parte. Hoje em dia, a gente tem toda uma equipe técnica da Enterprise focada em dar o suporte a respeito disso. Então, somos em 3 pessoas, eu, especialista de bioinformática, e 2 especialistas de produtos, capazes de ajudar vocês com a escolha do kit. O que eu quis trazer aqui, principalmente, são exemplos dos kits que a gente já tem comercialmente disponível. São 2 principais kits, o kit rápido, esses daqui, que são focados em um preparo de biblioteca mais simples, mais rápido, com baixa estrutura, e o kit de ligação, que é focado em um preparo um pouco mais complexo com enzimas adicionais e focando num rendimento maior. Eventualmente, se vocês tiverem dúvida a respeito da escolha do protocolo, esse tipo de coisa, a gente sempre pode entrar em contato para entender a demanda de vocês e sugerir o melhor kit. O que é importante destacar é os protocolos padrões que a Nanopore já tem disponível, eles são básicos. Eles são essenciais para vocês começarem a tecnologia. Mas existem diversas etapas de otimização do processo, otimização de protocolo, que otimizam muito o output e a entrega de resultados. E essas otimizações, por mais que sejam extra-oficiais, são otimizações que a gente, como equipe de suporte, como equipe de atenção, consegue passar para vocês. Um exemplo disso é esse protocolo, que foi publicado em janeiro desse ano, focado para sequenciamento metagenômico de patógenos bacterianos, virais e fúngicos. Ele é um workflow extremamente otimizado em etapas de banho, etapa de fragmentação, etapa de centrifugação, para a gente conseguir esse maior output, de fato, responder à pergunta biológica. ele permite multiplexação de até 24 amostras dentro desse contexto. Aqui eu trouxe para vocês, de fato, como funciona o sequenciamento em si. Então, o sequenciamento, ele acontece por nanoporos, que são proteínas transmembranas que dão, de fato, nome à tecnologia. Esses nanoporos, eles ficam ali em uma membrana de suporte que está passando uma corrente elétrica nessa membrana. Quando a gente fala preparar uma biblioteca, o preparo da biblioteca envolve a adição daquela enzima motora à extremidade da molécula de DNA que vai guiar o fragmento para passar através desse poro. À medida que o fragmento passa, a gente percebe aqui na metade do poro uma constrição que vai fazer a leitura dessa base. E como que essa constrição faz ler a base de fato? Eu tinha comentado para vocês antes que tem essa corrente elétrica passando aqui na membrana. Quando a base passa por essa constrição, ela provoca uma variação na corrente elétrica característica da base em questão. Em outras palavras, a adenina vai ter um padrão específico, a citosina outro padrão e a citosina metilada outro padrão. E é assim que a tecnologia consegue, no mesmo sequenciamento, entregar informações genômicas e epigenômicas de bases modificadas de metilação para a gente. O sequenciamento ele acontece ali naquela matriz orgânica que está dentro de flow cells. Flow cells são células de fluxos que a gente vai aplicar à biblioteca para correr a amostra. A gente tem dois tipos de flow cells disponíveis comercialmente, as de Minion e as de Promethyon. Essencialmente, elas são muito parecidas. Elas vão entregar a mesma qualidade, a mesma acurácia. A única diferença é o rendimento, o quanto que essas flow cells vão produzir de dados para a gente. A de Minion é focada no rendimento menor, mais baixo, para poucas amostras, bactérias e vírus, enquanto a de Promethyon para rendimentos maiores, sejam de organismos mais complexos ou uma maior quantidade de amostra. Cada um desse grupo de flow cells vai ser compatível com equipamentos específicos, sendo a de Minion o MK1D e o Gridiron, e a de Promethyon o P2 solo, o P2 integrado e o P24. Nesse caso, o MK1D e o P2 solo são dispositivos de entrada que não possuem módulo computacional acoplado. Então, a gente precisa fornecer um computador como parte do processo de sequenciamento. Mais uma vez, retomando aqueles 3 pilares que eu comentei, sequenciamento por qualquer pessoa, pensando nessa questão de acessibilidade, eles deixam essa customização por parte do usuário por uma questão de, às vezes, não ter tempo, não ter verba, não ter infraestrutura para conseguir sequenciar em campo. Então, essa questão de sequenciamento de entrada já ajuda a acessibilizar e democratizar o acesso à tecnologia. No caso do MK1D, ele foi a evolução que a gente teve para o modelo de entrada anterior, que é o MK1B. Ali fora a gente está com um exemplo dele, então quem tiver interesse, alguns já passaram, mas se alguém tiver interesse em conhecer, a gente tem com uma carcaça dele um exemplo de demonstração que vocês podem ver as principais diferenças que ele teve. Tá? Como principal diferença, eu posso destacar LEDs aqui na parte superior, que permitem monitorar o status da corrida, compatibilidade com USB-C, já visando compatibilidade com iPads, tablets e computadores futuros, e, por último, a principal mudança que teve foi a estabilidade térmica, que aumentou de 10 a 35 graus por conta de uma mudança no método de refrigeração. Antes era baseado em uma ventoinha e agora virou um Peltier. Isso melhorou muito o range de atuação dele, principalmente para quem trabalha em campo. Com isso, então, a gente tem a parte de sequenciamento, e o sequenciamento vai acontecer ali no computador por um software chamado MinKnow. Esse software ele vai coordenar todo o processo de sequenciamento, pegar os dados que estão sendo gerados ali pela tua flow cell, as diferenças de padrão elétrico que são identificadas e converter a bases, que é a informação que a gente precisa. Esse processo de conversão é chamado de base calling, e ele é feito pelo Dourado, que é um software que usa bastante GPU, para conseguir fazer esse processo. Esse processo, então, idealmente, no final de um sequenciamento, a gente teria os POD5, que seriam os arquivos de dado bruto do sequenciamento, e o FASTQ e o BAN, que são arquivos de input que a gente poderia usar para workflows de análise. Com isso, naquelas 3 etapas iniciais, preparo, sequenciamento e análise, eu já dei uma introduzida sobre o preparo e sequenciamento. E agora a gente entra, de fato, na parte de análise, que eu acho mais interessante. Lembrando, todos esses softwares são gratuitos e eles são super amigáveis de usar e práticos de instalar. No caso do App2Me, ele possui uma interface de usuário, é um aplicativo que a gente baixa no nosso computador, pode rodar localmente, pode rodar em nuvem, tem hoje em dia mais de 15 workflows diferentes para as principais demandas e aplicações. Todos os workflows são em códigos abertos e eles permitem com que a gente consiga customizar, eventualmente, um pipeline interno, se tiver necessidade de deixar algo mais específico e personalizado para a aplicação de vocês, é possível, porque tudo é em código aberto. Vocês têm a possibilidade de rodar em linha de comando e na nuvem, e para instalar é bem simples também. Só no caso do Windows, precisa configurar um ambiente intermediário chamado WSL, que é um ambiente que vai garantir a compatibilidade do Linux para o Windows. E no caso de Ubuntu e macOS, o Docker, que é super fácil de instalar, em questão de 3 linhas a gente já consegue. Dentro desses 15, aproximadamente, workflows que a gente tem disponível, eu quis trazer para vocês alguns do que eu acho principal para a gente conseguir, de fato, conduzir uma pesquisa com vigilância genômica. Esses seriam o de metagenômica, metabarcoding, isolados bacterianos e alguns exemplos de doenças infecciosas. No caso de metagenômica, como é a etapa que a gente vai conduzir, então já fez o preparo, a gente fez o sequenciamento e conseguiu produzir aqueles arquivos POD5 e FASTQ. A gente vai pegar esses arquivos, carregar dentro do workflow que a gente acabou de baixar dentro do aplicativo e disparar a análise. Uma vez que a gente dispara, tem diversas métricas, parâmetros que a gente consegue fazer para ajustar e conseguir um melhor resultado final. Esses parâmetros não têm default, eles são definidos a critério de cada usuário final para conseguir entregar o melhor resultado de fato. E o que a gente espera ver no caso de metagenômica? O workflow de metagenômica ele é constituído por 2 ferramentas de busca que vão pegar as nossas reads geradas pelo sequenciamento e bater em bancos de dados específicos, sendo essas ferramentas o Kraken 2, uma ferramenta mais lenta e menos precisa, e a Minimap 2, que é uma ferramenta um pouco mais robusta e com uma melhor qualidade. Em relação aos bancos de dados, por default, a gente tem 6 bancos de dados já instalados, mas isso não limita vocês de instalarem ou customizarem, carregarem bancos de dados próprios, não tem problema. É só que, visando essa questão de praticidade e de facilidade do usuário, já tem esses workflows muito bons do NCBI e Kraken 2 pré-carregados. No final, o que a gente espera obter? Todos os workflows do App2Me vão soltar um relatório HTML, que é no formato como se fosse um PDF, compilando um resumo das principais informações que foram encontradas ali. Nesse relatório vão ter gráficos visuais, interativos, que vocês podem exportar e usar como preferirem. No caso do de metagenômica, o principal deles é esse gráfico que a gente vê aqui, que ele começa com as classificações taxonômicas, indo desde superreino até nível de espécie. Então, de fato, é uma tecnologia que, seja abordagem de metagenômica, seja outras abordagens que a gente vai ver, consegue te entregar da amostra à resposta em nível de resolução de espécie. Outras 2 funcionalidades bem interessantes desse workflow é que ele permite que vocês, por bioinformática, removam, depletem o genoma do hospedeiro que possa vir contaminante daquele material. Recentemente, eu estava conversando com um grupo de pesquisa que comentou que trabalha com microbiota humana, e de 50% a 80% do material genético que eles possuem no rendimento da Flow Cell é de DNA humano, que não é do interesse delas. Isso seria uma forma de depletar por bioinformática, mas a tecnologia também conta com uma estratégia chamada Adaptive Sampling, que permite com que você deplete antes, na Flow Cell, ali, durante o sequenciamento, você já faz a depleção, faz o enriquecimento da tua região de interesse e economiza os teus poros para um melhor rendimento. Muito similar ao de metagenômica, a gente tem um workflow de 16S. Eu gosto de chamar ele de metabarcoding, na verdade, porque ele não mira só no 16S, mas ele pega 16, 18, 28 e ITS. Nesse momento, ele só não pega COI, que é um dos principais barcodes que as pessoas também gostam de usar. Mas ele consegue... A ideia é a mesma da metagenômica. 2 ferramentas de busca, Kraken e Minimap, que vão consultar bancos de dados específicos, do NCBI ou do Silva, para trazer um nível de classificação taxonômica chegando a nível de espécie. Ele também permite a depleção do sistema hospedeiro. E a única coisa adicional que o de metagenômica tem, que ele não tem, é a detecção de genes de resistência a antimicrobianos. Um outro workflow muito bacana é as pessoas que trabalham com metagenômica, que vão fazer investigação microbiológica, às vezes costumam usar uma abordagem de metagenômica e barcoding para fazer uma triagem inicial. E uma vez que elas conseguem identificar e classificar a amostra que tem ali, elas partem para uma abordagem mais isolada, fazendo o cultivo e isolamento daquela bactéria para chegar numa melhor caracterização e montagem. Esse workflow ele faz parte de um pacote completo que a gente tem desde o preparo, sequenciamento, até a resposta, passando por vários passos de otimização para entregar o melhor resultado. Esse protocolo ele chama No Miss. É um protocolo da Nanopore voltado exclusivamente para isolados bacterianos. Uma vez que você já tem a parte do preparo e sequenciamento, o que vocês esperariam obter dele aqui na análise, no final? Clássico relatório HTML, que todo workflow volta, mas, além disso, ele volta a arquivos adicionais que não são possíveis de colocar no relatório, como o arquivo FASTA da sua montagem, que foi solicitada, um arquivo GFF da sua anotação e um VCF da chamada de variantes. Além disso, ele tem uma opção adicional chamada Isolates, que permite com que você desloque o workflow para 3 etapas adicionais que vão auxiliar na sua análise. Quais seriam essas? Primeira, classificação taxonômica por tipagem multilocus, por MLST. Segundo, identificação de genes de resistência antimicrobiano, pelo ResFinder e o Abricade. E o último, mas esse é mais para a salmonela, é a sorotipagem da salmonela na identificação de espécie. Tudo isso vocês conseguem obter só com esse workflow, sem nenhum preparo adicional. E os últimos workflows que eu queria trazer aqui para vocês, por uma questão de tempo eu não trouxe eles isoladamente, mas só para mencionar que são possíveis, eles existem, são os de doenças infecciosas. Nesses a gente tem 4 principais, que são o SARS-CoV, o influenza A e B, o de tuberculose bacteriana e de monkeypox. Acredito que alguns de vocês já tenham ou tido contato com SARS-CoV, processamento de SARS-CoV, ou conheçam algum grupo de pesquisa que tenha. Naquela época, existia um pipeline que ia desde a parte de preparo até a de análise, chamado ARTIC. Esse pipeline foi intensivamente utilizado e validado e, posteriormente, incorporado nesse workflow do App2Me como uma forma de facilitar o processo de análise. Esse é o workflow que a gente tem para o SARS-CoV do ARTIC. No caso de influenza A e B, a gente tem um workflow também que permite a montagem chamada de variantes dos vírus de influenza A e B, dos 8 segmentos, daqueles 8 fragmentos, e ele pode ser aplicado tanto para a gripe humana, quanto suína, quanto aviária, não tem problema. Um ponto curioso a respeito disso. Uma parte que eu gosto muito do trabalho que eu faço é que eu tenho contato com diferentes grupos de pesquisa do Brasil inteiro. E isso a gente conhece um pouquinho da pesquisa de cada um. Recentemente, eu conversei com um grupo de pesquisa que estava interessado em usar a Nanopore para monitoramento de patógenos virais respiratórios em animais marinhos. E eu, em toda a minha ignorância, cheguei para ela e perguntei, mas o golfinho tem gripe? Ela falou, não só tem, como morre. Parte do grupo de pesquisa deles consistia de uma menina que ficava viajando de navio em parceria com outros grupos de pesquisa estrangeiros para monitorar o índice de mortalidade na região antártica desses animais marinhos. Eles acabaram percebendo que aves migratórias estavam trazendo esses vírus de influenza A e B para aquela região e causando um alto índice de mortalidade. Eu saí um pouco fascinado, um pouco assustado com essa informação, mas é um ponto que eu sinto muito enriquecedor de trocar essa experiência com cada usuário e conseguir ajudar vocês. E isso é uma outra informação que é muito importante deixar claro, que a gente está aqui no Brasil para representar a tecnologia, mas para ajudar vocês a conduzirem a pesquisa de fato. Então, mesmo que a gente não tenha as respostas diretamente para ajudar, a gente tem contato com a Nanopore e a gente tem contato com o Brasil inteiro. Então, a dúvida que um de vocês possam ter, outros também podem. A tecnologia da Nanopore, o crescimento da empresa foi muito baseado em comunidade, em compartilhamento de informação, em diversidade de análise, e é esse o papel que a gente também executa aqui no Brasil. Claro que existe toda uma proteção de dados, uma questão de LGPD que a gente prioriza frente a esse compartilhamento. Mas é importante saber que vocês têm esse meio de comunicação entre diferentes grupos aqui no Brasil. Voltando, a gente tem o de tuberculose bacteriana, que é voltado bem rápido para a classificação de genes de resistência antimicrobiano, principalmente em ambientes hospitalares, e, por fim, o de monkeypox, que ficou muito famoso por conta dos recentes surtos que a gente teve. Então, já dei uma breve e rápida introduzida aqui para vocês a respeito de preparo, sequenciamento e análise. Mas, de fato, qual que é o impacto disso na vida real? O que a gente pretende ver com essas otimizações e como que a Nanopore consegue entregar uma boa ferramenta para a vigilância epidemiológica? Aqui eu trouxe alguns exemplos para a gente conseguir identificar. O primeiro é uma série de estudos voltados para isolados bacterianos, onde eles conseguiram identificar um altíssimo ganho na acurácia com os mais recentes avanços e atualizações que a gente teve da tecnologia. Então, são muitos grupos de pesquisas trabalhando com diferentes tipos, famílias de bactéria, que reportaram uma qualidade acima de 20 para o sequenciamento e uma qualidade de consenso de montagem acima de Q60. Q60, dentro da escala Phred Score de sequenciamento, é virtualmente um erro a cada 1 milhão de bases. Isso é o que a gente espera de acurácia para consenso, para essa maioria de famílias de bactérias que foi validada ao longo do tempo. Isso se deve principalmente a alguns avanços que a gente teve na flow cell. Então, quando eu tinha mostrado para vocês aquele esquema inicial de como o sequenciamento acontece, eu mencionei aquela uma constrição que tinha dentro do nanoporo. A flow cell nova, a R10, para quem já está acostumado, ela tem 2 constrições que fazem com que a mesma base seja lida 2 vezes. Esse foi um dos motivos da gente ter um ganho na acurácia. E o segundo, e também extremamente relevante, foram diversas atualizações de software, para a gente conseguir um base calling, um modelo de chamada de bases com altíssima acurácia. Um outro exemplo que eu trouxe aqui para vocês de aplicação real é como a gente tem, em um cenário global, a nanopora ajudando com essa parte de monitoramento de vírus, vigilância patógena e epidemiológica. Então, aqui a gente percebe, desde 2015, lá, ebola na África, indo para 2019. Aqui no Brasil, em 2018, a gente teve um estudo muito bacana sobre febre amarela, com participação do Dr. Luiz Alcântara, que falou aqui também, e vários outros estudos ocorrendo simultaneamente desde aquela época até hoje, como gripe suína, identificação de genes de resistência antimicrobiano e outras infecções. E aqui um pouco atrasado para a Semana do Meio Ambiente, mas eu quis trazer um exemplo para vocês de monitoramento de biodiversidade usando a tecnologia nanopore. Então, esse é um pássaro da Nova Zelândia, classificado como criticamente ameaçado de extinção. E no país existem diversas políticas públicas que realmente visam a proteção desse pássaro. Mas, eventualmente, dado o baixo número deles, eles precisam revisitar essas políticas para ver se elas estão sendo efetivas ou precisam ser mais incisivas. Um grupo de pesquisa acabou percebendo que, com o monitoramento de DNA ambiental a partir de 1 grama de solo superficial, eles conseguiram não só rastrear o movimento desses pássaros naquela região, como também estimar uma quantidade dos indivíduos. Esse é um estudo muito bacana que foi financiado, por uma iniciativa que a Nanopore tem chamada Org-One. A Nanopore ela trabalha com diversas iniciativas. O Ep2Me que eu mostrei para vocês, apesar de ser um aplicativo, ele é uma iniciativa de facilitar o processo de análise de dados por qualquer pessoa. E essa iniciativa da Org-One, ela visa a preservação da biodiversidade. A Nanopore ela financia a tua pesquisa como um todo e você encaminha para ela os dados do seu sequenciamento, principalmente para espécies não descritas e não caracterizadas. E, por fim, falando em iniciativas e programas e bolsas, esse ano teve uma atualização muito bacana da Nanopore para conseguir ajudar as pessoas ao longo do planeta. A cada trimestre, eles estão fazendo uma seleção de melhores projetos para conseguir financiar. É o que chama Oxford Nanopore Grant Program. No que ele consiste? Eles abrem inicialmente a cada trimestre e você submete um projeto. Esse projeto vai ser avaliado dentro de 3 meses e, se for agraciado, você tem a opção de receber ou um pacote de sequenciador voltado para MK1D ou um pacote voltado para P2 solo. São 30 para cada uma dessas 2 categorias, só que é em cenário global. Atualmente, a gente está fechando a 2ª rodada de classificação, mas na primeira rodada que a gente teve aqui no Brasil, 2 pessoas foram agraciadas. Foram 2 grupos de pesquisa, um para o MK1D e um para o P2 solo. E o único requisito é ter uma pesquisa com grande relevância social e científica. Basicamente isso. Então, quando a gente olha no micro, realmente, todo esse ganho que a gente teve na acurácia, ser uma tecnologia que trabalha com long reads e uma velocidade rápida de processamento, ajuda muito no processo de vigilância. Mas, olhando no contexto macro, todo esse compromisso que a tecnologia tem com entregar para a gente um aspecto e iniciativa social, científica e ambiental, fazem dela ser um bom pilar para a vigilância genômica. E é por isso que a gente consegue, através de todo esse compromisso, entregar um sequenciamento de qualquer coisa, por qualquer pessoa e em qualquer lugar. Obrigado.
Obrigado. Alguém tem alguma dúvida? Ok.
Obrigado,
Obrigado.
pessoal.
Okay, so we can get back to the program, trying to correct the changes we already had. So we'll now have Lisa Antonelli from Fiocruz-Minas Gerais. Thank you, Liz. Vocês colocam um slide em cima, por favor, da Liz. Aí. Obrigado.
So, good morning, and thank you, Ricardo, again for the invitation. And today I'm going to talk about asymptomatic Plasmodium vivax infection and how these individuals, not patients, what they are trying to hide from us. So malaria needs no introduction, especially after Irene's talk from yesterday. But I'd like to make a point here that although the number of Plasmodium vivax cases here is an order of magnitude lower than the number of malarias caused by Plasmodium vivax, there are several reasons to study Plasmodium vivax. So the first one is that 80% of the cases we see in the Americas are caused by Plasmodium vivax, and most of them occur in the Amazon. And there are also a lot of particularities of Plasmodium falciparum that make the control and the elimination of this parasite a challenge. So I'm going to go a little bit quick here, but vivax parasites has the ability to release, to relapse from long-lasting dormant liver stages. They actually, gametocytes are produced very early during their development, and this increased the high transmission potential because actually the gametocytes appear even before the symptoms. They have high infectivity. They are highly infectivity to mosquitoes. They have shorter development cycle in the vector host compared to the other Plasmodium. And I think this is an important thing that actually their resilience made them to adapt to lower temperature, thus extending its distribution in the host and also into temperate regions. So we know less than we know about falciparum, but here we know that vivax, actually the symptomatic malaria vivax, it's a febrile syndrome and has this this particular pattern here. So we know when we're going to have less cases and more cases. But how about the asymptomatic infection? And just to make more clear here, the asymptomatics are the ones that are infected, but they don't show any symptoms, or they don't show the symptoms that we are looking for or that make them to look healthy care. So epidemiologically, how the asymptomatic Plasmodium vivax infection behaves and why it is important. And malaria, actually all kinds of malarias are being represented by an iceberg. So what happened here is we can see more than 200 million cases of symptomatic malaria a year. However, 5 times more people infected with Plasmodium don't show anything. They're asymptomatic. And this is the problem, right? Because you cannot fight an enemy that you cannot see. So this project actually started a little bit earlier than 2018. And it involves a lot of different PIs. And in the immunology part, it counted with Ricardo Gazinelli, Douglas Golenbach, and myself. So our hypothesis was that asymptomatic Plasmodium vivax infected individuals developed a robust immunity that maintains low parasite biomass, preventing systemic inflammation and reducing vector infectivity. So to do so, actually what we wanted was to identify immune biomarkers of resistance and susceptibility to disease, as well as mosquito infectivity, to better understand malaria pathogenesis. And to do that, we already worked on this endemic area here in Porto Velho with symptomatic malaria, and we started to work with asymptomatic malaria in Candeias do Jamari. So Candeias do Jamari is here, and actually was a perfect place to start this project. This was like very smartly chosen by Ricardo and Douglas. And this municipality has around 27,000 inhabitants, and it has this river that around the town, and there's a lot of forest as well. So since asymptomatic doesn't come to us, what we had to do was to do a kind of census. So we're going to their homes, like the homes of the community, knocking door to door, and we asked for some blood, drop of blood, and we did some PCR and microscopy here. So the ones that were positive for vivax infection, we invited to participate in this study, and we invited them to go to Porto Velho, where they were seen by a clinician. They answered a lot of questions, and we were taking some blood from them. So at the same time, in Porto Velho, we were recruiting the symptomatic patients and also the non-infected ones. And with the samples, we did a lot of things, and we're still doing a lot of things with the samples. And this is the team. So the team started actually with 4 people, so it was really tough time. And 2 years later, we were with a lot of people working. And I have to thank a lot Greg and Camila that helped organizing everything and doing all the experiments. So this is Candeia, and what we saw here was that actually we had 3 different areas. The 2 first ones were close to the river, and it is showing in red here that we had more asymptomatic infections in these 2 areas, and we had also more symptomatic infection here. The 3rd area, further from the river, we had less asymptomatic infection and less symptomatic infections too. So what we saw as well, and was what we expected here, that asymptomatic infected people had much lower parasite copies compared to the symptomatic. And here is an interesting result as well, because we were doing 3 PCR for each sample, and the ones that were consistent positive had more copies than the ones that had 1 or 2 PCR positive. And we did, so we carry on to do this membrane feeding assay because we're interested in seeing if the asymptomatic, even having low parasite loads, they were able to infect mosquitoes. So what we did here, we did this membrane with Maísa, and actually in this graph we showed that 9 blood of symptomatic people infect mosquito. And half of the blood from asymptomatic also infected the mosquito. So, and infected less mosquitoes. So, and we can see here as well that I don't know if you can see here, but this is the midgut of the mosquito. This is Anopheles darlingi. And we see much more oocysts in the midgut of the mosquitoes that were fed with symptomatic blood when compared to asymptomatic. This I'm gonna jump. And then Greg came up with this really nice cartoon here. So the model is that the symptomatic have high parasitemia, but they are diagnosed, treated, and then what happened is that the transmission stopped. But when we have asymptomatic, we have this fluctuant and also low parasitemia. And this parasitemia, since these patients are not treated, we have this, this low parasitemia infect mosquito that lasts longer. And how about the immunosignature of these patients? So I'm not going to talk about all of this, but here, based on what we studied before in symptomatic infections and also based on the literature, we chose approximately 350 features to analyze. And I have to say this is in a protein level, right? So we looked for antibodies, cell frequencies, cytokine, chemokine, growth factors in symptomatic, asymptomatic, and actually, for the sake of the time, I'm going to talk about the cell phenotypes, and more particularly, I'm going to talk about T and B lymphocytes. So what happened with T lymphocytes? We described before that with a lot of cytokines, a lot of inflammation, there's a lot of upregulation of activation and inhibitor molecules on these cells in symptomatic. However, we didn't see any upregulation in the asymptomatic here comparing to symptomatic. They are pretty much as the controls. And if you observe here, actually the asymptomatic had even lower frequency of cells expressing some of the markers that we looked compared with symptomatic and asymptomatic. And when we looked for the T helper compartments, the TH cells, what we saw here, and we looked based in the expression of chemokine receptors, we saw that asymptomatic had more frequency, had expanded TH1 cells comparing with symptomatic and also with controls. And we looked the other TH, and although no difference were found here in the T follicular helper cells, when we look the TH1 subset from this population, we see that both symptomatic and asymptomatic had higher frequency of these cells compared to control. And knowing that T follicular cells help the maturation, development of B cells, and also the antibody production, we look for the B cells. And you can see here in this T-SANI and in this box plot that if you look for plasma cells, actually only symptomatic have higher frequency of the cells compared to asymptomatic and controls. We don't see any difference in this. We don't see any difference in asymptomatic when we look at atypical memory cells, the ones related with TH1 response because they express TBET, and we also didn't see difference among controls and asymptomatic. However, we see that symptomatic have less frequency of this classical memory B cells compared to asymptomatic and control. However, if we look the proliferating cells, we see that atypical memory cells, the one related to TH1 response, is different when we compare symptomatic, asymptomatic, and control here. And we also see that the higher the frequency of these atypical cells proliferating, this is in the case of asymptomatic, we have higher parasitemia and we have high levels of antibodies against AMA1 and MSP1 here. So we did this sparse partial least square discrimination analysis, and this actually represents all the markers, all the molecules that we analyzed, the 350 markers, and we can see here that this latent component 1 can discriminate symptomatic compared to the other two groups, and latent component 2 can discriminate asymptomatic and controls here. And we can see here in this ROC curve that the accuracy here is good. And if actually, if we look the contribution of each of those actually not markers or the features, we see that the ones that have the associated with TH1 phenotype, they contribute in this latent component 2 to discriminate the asymptomatic. So the take-home message here first is how asymptomatic behave here, like more epidemiological part of the study that we have to determine very well to be able to work with this population. And for the second time, we had that asymptomatic malaria is associated with the expansion of TH1 cells in atypical B cells contemporaneously with the absence of systemic inflammation. So our findings suggest an immunological signature induced by interferon gamma in Plasmodium vivax infected asymptomatic individuals. So I'd like to thank all the collaborators here and also the financial support. Thank you.
Thank you very much, Liz. We have time for questions. I can start. This, to me, was very surprising that you really have a difference in parasitemia between asymptomatic and symptomatic, which is the opposite of what we see in viral infections, where asymptomatic and symptomatic have basically the same viral titers, but what differentiates is the immune response. So can you elaborate on that? Why do you think that's...
Oh, actually, I'm not sure. That was what I think everybody expected for parasites, at least for malaria, because when you think about falciparum, is the same. In very highly endemic area, kids can have high parasitemia and be asymptomatic, but in our case here that we are low endemicity country, we don't see any high parasitemia associated with asymptomatic status.
Alan? Yes.
It does. Wonderful. Great talk, Liz.
Thank you.
So I just don't know the field that well, but is there any thought that perhaps these asymptomatics are contributing to transmission? You've shown that you can actually infect mosquitoes from them. So do you think one would want to get rid of the malaria cases and the asymptomatic cases to help reduce the spread of Vivax?
Yeah, I believe so. But we don't have data, actually. We know that because the problem is that the area we work, when you have increase, the areas with higher asymptomatic cases, we have also symptomatic cases. So we are not sure who is transmitting malaria. But actually, I was almost certain that actually, we're doing a great thing here. Then David Sachs told me they did the same for leishmaniasis. And when they did this in the laboratory, as we did, they found that they could transmit leishmaniasis during membrane-filled assay, but then they went to the field, and it was not the same story. So I'm not sure. We don't have data, and there's nothing published yet.
Because for like with TB, you'd want to treat those asymptomatics.
Oh
'Cause
yeah,
they're
and
a
actually
source
this
of-
this is something that
Yeah.
changed while we were
Yeah.
not because of us, but because of the pandemics. Because before we were not able to treat these patients because the health ministry didn't
okay.
recognize as a diagnosed test. But after COVID, we started treating these patients. And before they didn't want to be treated, and now they want the treatment. So it was huge, like, change, I think. Let's see.
Santuza.
Hey, Liz. Beautiful, beautiful work. Thank you.
Thank you.
Yeah, I was going to ask 2 questions, but the second one you just answer, because if the point is to treat the asymptomatic or not, which is, I guess, that's what needs to be done. But I think the main point is diagnosis. How
Yeah.
I don't remember how you showed that you identified the asymptomatic was
By
slide?
PCR.
By PCR.
They are negative.
So there was no way you can do PCR in big areas.
No.
So are you going to comment on rapid test, maybe?
Well, I'm not sure how are the results, but there's a test that we started.
Yeah, I
Julia-
think one of the main points is if you do by PCR, as she showed, ups and downs. So if you do a transversal cut, so a lot of them will be negative. So what we're trying to do now is do both the rapid test and the PCR. But you have a problem because some of the people that are not infected will be positive on the rapid test. So you have to have a follow-up anyway, but I think the rapid test will help to get a lot of the asymptomatics that are not being PCR positive. So we're trying to tune the rapid test that's more accurate to define what's asymptomatic or not.
Right. Thank you.
Well, I have a suggestion. You know, in Southeast Asia, they do diagnostics, feeding mosquitoes on people, and then they wait and test the mosquito. So you could.
But this
But
actually,
not membrane
this is
feeding.
really.
It's direct on human feeding. Huh?
Ah,
Yeah,
yeah.
it's a lot more efficient than if you take the blood and you give the.
A lot of them will be, I suspect that will be not infected.
Yeah, not infected.
Will be infected. It's
Well, thank you very much, Liz.
Thank you.
Okay, we can move on now to the next speaker, Elton Santiago from the Department of Biochemistry and Immunology here at UFMG. Thank you, Elton.
Thank you, João. I would like to thank the organizers, Ricardo and Luis, for the invitation to present my work on dengue. Dengue, everybody knows, is a big problem of public health with estimation of 390 million cases per year, of which 100 million are symptomatic, leading to 20,000 deaths worldwide. It's a tropical disease, but because of climate change, it's invading the subtropics, and currently it is estimated that about 50% or 40% of dengue in some areas of Brazil might be due to climate change. Dengue is classified as dengue without warning signs, with warning signs, and severe dengue by WHO. So in our work, we just use dengue warning signs negative, which is a mild case of dengue, or dengue with warning signs positive and severe dengue combined, because we didn't have enough severe cases in our cohort to separate this as a group. The clinical evolution is that the dengue presents fever, while there is viremia. But when the viremia is resolving, and that happens when the antibody level starts to rise, the individual may have drops in platelets and increased hematoct, which is associated with, may be associated with shock, bleeding, and organ impairment, which is the severe dengue, or what was known before as hemorrhagic dengue. So to clear viruses, usually when you get infected, the virus starts proliferating, and then the innate immunity is immediately activated, but the innate immunity cannot clear the virus. Usually it decreases the proliferation of the virus, and the virus is really cleared when the adaptive immunity kicks in. So the virus is cleared, then the innate immunity is deactivated, and then you have a memory cells after the infection is cleared. When you have a second infection, the virus starts proliferating, the innate immunity is activated, but because you have memory, the adaptive immunity kicks in faster and stronger and eliminates the virus, turning off the immune response. But to understand why some people develop severe dengue or mild dengue, we also need to understand these two concepts of tolerance and resistance to infection. So usually you are happy living your life, but you can get an infection, the pathogen proliferates inside the body, and then you get not so happy. But as the pathogen proliferates more, usually you get a worse health status and get very sad. So in order to control this situation, you need to be resistant to the infection. So, and impair the proliferation of the pathogen. But some people, the pathogen proliferates in the same way as some other, and some just stay well. Lisa just talked about the asymptomatic, although they have less parasites. But some people with the same pathogen load, they stay well, while others stay very bad. So, and that's because of tolerance to the infection. Individuals that usually have higher tolerance, they can support higher loads of parasite without feeling too bad. So the thing is that you get an infection, the infection causes you pathology, which prevents your homeostasis and make you sad. Then your immune system reacts with a pro-inflammatory reaction, which fights the infection and brings you back to homeostasis. However, if you have low tolerance to the infection, this pro-inflammatory can itself cause pathology. So to prevent that, the immune system has an anti-inflammatory reaction that operates at the same time, preventing immunopathology. So to investigate how some individuals would develop severe dengue or dengue with warning signs, we had two hypotheses. One is a compromised effector response. It means that the individuals would not mount the appropriate pro-inflammatory response, and that would allow pathology because of the infection. And the other hypothesis is that individuals may mount an appropriate pro-inflammatory response, but would have a compromised regulatory response that would cause immune pathology. So we decided first to investigate one of the thought mechanisms of dengue complication, which is the cytokine storm that would kick in this critical phase. So I forgot to emphasize that there are these three phases of the disease: febrile, critical, where the complications happens, but the viremia usually is gone, and then the recovery or convalescence phase, when everything is fine. So we got blood from individuals with dengue during the critical phase, and we evaluated interferon gamma, which is and TNF, which are cytokines associated with resistance to fight the infection. And individuals developing mild dengue would have higher levels of interferon gamma and TNF when compared to individuals developing dengue with warning signs. But the level of IL-10 was similar. So this was indicating that the resistance of individuals developing severe dengue might be compromised. So we decided to check the sources of these cytokines, and we went to CD4 and CD8 cells during critical or convalescent phases. And we found that individuals with mild dengue, they have more CD4 and CD8 producing TNF or IL-10, either in critical phases, sometimes in the convalescence here for IL-10. We also investigated double producer cells, which are multifunctional T cells. So we found this very interesting population producing interferon gamma and IL-10 at the same time, present in individuals developing mild dengue, both during critical and convalescence, and in the CD4 compartment and also in the CD8 compartment. And we also found this very interesting population of CD8 producing interferon gamma and TNF, which are very potent inflammatory T cells. And we also found cells producing the three cytokines, both, especially CD8s, but CD4 here too in the convalescence, but CD8s in the critical and convalescent phase producing the three cytokines. So this was suggesting, and these triple producing cells would positively correlate with the level of platelets, which is a marker of dengue complications. So individuals developing severe dengue, the platelets drop a lot, and we could see that these cells, multifunctional T cells, were associated with protection from this development. So far, it was being suggested that the severe cases of dengue display low levels of plasma interferon gamma and TNF, and present lower frequencies of dengue-specific CD4 and CD8, producing interferon gamma, TNF, and IL-10, especially double or triple producers, which was suggesting that severe cases of dengue were associated with impaired T-cell adaptive immunity, so they do have some level of compromise in the resistance to infection. So we decided also to check the other hypothesis about the compromised regulatory response, and we decided to evaluate Tregs, which are the master regulations of peripheral tolerance, and Tregs employ several mechanisms to regulate immune responses, including the production of IL-10 and several receptors, inhibitory receptors like GITR and CD200, which I'm going to show in a bit. And the level of Tregs in the infection usually is not altered, but in the individuals developing dengue with warning signs seems a little bit decreased in the critical phase, which returns to normal in the convalescence. And these Tregs they do express less GITR. In the individuals with severe dengue express less GITR and less CD200 when compared to individuals developing mild dengue. On the other hand, individuals with developing severe dengue seems to express higher levels of PD-1 by the Tregs, which is associated with Treg dysfunction. But most importantly, we stimulated the PBMCs with dengue antigens. We used two antigens, envelope and F3, and we found that the individuals developing mild dengue, they have a strong population that responds to these antigens producing IL-10. These are Tregs producing IL-10, while individuals that develop severe dengue does not have this population. Here we can see the increase in the envelope specific, and here in S3 specific Tregs, producers of IL-10. Marcela also evaluated the capacity, compared the capacity of the CD40 cells, which are not Tregs, to produce IL-10, and she compared that with the Tregs, and the Tregs, not surprisingly, produce way more IL-10 than the TR1 cells. So these individuals do have also a compromised tolerance to dengue. So that gave us a problem, because in the literature, there was this data here showing that individuals that develop hemorrhagic dengue fever, it was a late 90 paper where the classification was different. And in this paper, Sharon Green showed that individuals that develop hemorrhagic dengue fever, they have higher levels of interferon gamma in the circulation before the defervescence. In my data, I evaluated after the defervescence, when the critical phase starts. So, and what I found is that, sorry, my throat is a little sore today. But what I found was that individuals that developed hemorrhagic dengue would have compromised, thank you, Jean, would have compromised effector immunity. But here, the data of Sharon Green shows higher levels of interferon gamma in the plasma. So we decided to check what was happening here. So, and our hypothesis was that this interferon gamma that Sharon Green observed was coming from innate immunity. So we went to check innate lymphoid cells and NKs during the febrile phases, during the febrile phase of dengue. And here is our results. Here, in these graphs here, I have the CD3 in this axis and interferon gamma in this axis. And we can see here individuals that are not infected, you can find very little cells producing interferon gamma. And most of these cells were CD3 positive T cells, and very few coming from the innate immunity. But in the individuals that have dengue, either without warning signs or with warning signs, the level of interferon gamma during the febrile phases producing interferon gamma in the CD3 negative increases a lot, and actually takes over like 30% to 75% of the, or even more, maybe 80% of the interferon gamma producing cells. So we decided to investigate which cells were these CD3 negative, interferon gamma positive, and we use 2 markers to identify ILCs, which is ID2, and EOMs to identify NK cells. Most of the cells didn't fall in each of those using these markers, but still we could see that the ID2 positive T cells were, and interferon gamma positive T cells, sorry, not T cells, interferon gamma positive ILCs were increased in individuals developing severe dengue or dengue with warning signs. On the other hand, NK cells were increased only in individuals that developed mild dengue. Because I didn't like this result too much, we decided to use another way to identify these T cells, which these lymphocytes, these innate lymphocytes, which is CD56 and CD127. So CD3 negative, CD56 positive usually are NK cells, while CD3 negative or lineage negative, CD127 positive usually are ILCs. So in the general number of them, the infection doesn't do much, but we can see that individuals developing dengue with warning signs, they have higher levels of ILCs producing interferon gamma, but now NK using this strategy was not different between the 2 clinical forms. And the ILCs of individuals with warning signs not only produce higher levels, I mean, they not only are in higher frequencies, but also they produce more interferon gamma per cell when compared to individuals developing mild dengue. So not only ILC1 or NK1, which are the innate lymphocyte that produces interferon gamma, was altered, but the severe dengue was associated with the immune dysregulation in this innate compartment. We can see that individuals with dengue with warning signs, they have higher levels of ILC2 that produces IL4, IL13, more IL-17, the ILC3s, and they also have more NK that produces IL-17. So the take-home message here is that if you get dengue and want to be happy, you need to have greater specific interferon gamma production in the febrile and critical phase, greater contribution of T cells in cytokine production, increased activation of multifunctional T cells, and increased Th1 reg, which are those T cells that produce interferon gamma and IL-10 at the same time. Here, Ricardo was one of the first to describe this thing, and it has now been described for many other diseases. And you want to have a balance, the specific and inflammatory process. Now, you don't want to do, if you get dengue, have lower specific interferon gamma production, or interferon gamma coming mainly from the innate immunity, lower activation of multifunctional T cells, lower activity of Th1 reg or Tregs, and increased, you don't want to have increased activity of ILCs and NK, which means higher unspecific inflammatory process. So these results suggest that the Th1 pro-inflammatory immune response balanced with the regulatory mechanism provided by, especially by TR1 and Treg cells, contribute to an appropriate resolution of infection, leading to better clinical evolution of dengue. This is the team that worked in this project, especially Marcela and Iracema, which did most of the experiments, and thank you so much.
Thank you very much, Elton. We have time for questions. Maybe I'll start, Elton, on on the T cell response to the antibody-dependent enhancement. Can you elaborate on how you connect this interferon gamma protective response, presumably by the innate immune response, and then the contribution of potentially of the antibodies on severe dengue?
Yeah, I don't see the innate immune response protecting that much. I see it causing more pathology in dengue. Now, how do I connect that with the antibody-dependent enhancement? What I believe is that the antibodies will cover the immature viral particles, lead to higher levels of proliferation of the virus, which will trigger higher innate immunity. And the thing is that because the individuals that develop the severe dengue, they have impaired T cell activation, the innate immunity will try to combat the infection. So what we are observing is that at some point, the adaptive immunity kicks in even in the individuals developing warning signs, but it takes longer. So while it takes longer, the person suffers more with the innate immunity that doesn't they synthesize cytokines just to everybody. I mean, to cause a lot of lesion while they are doing that.
Thank you. Professor, I don't know if you said at the beginning of your presentation, but did you check the patients for pre-existing dengue immunity? And the second, just a very fast one, quick one, is do you know which serotypes are involved? I mean, are different serotypes? Do you have only one serotype? And if you do, which one is it? Yeah. We checked the pre-immunity because we wanted to see if secondary was going to be different. 80% of our cohort was dengue positive at 7 days, which means IgG positive. It means that they were previously infected. Okay. So we didn't have enough to do a comparison, but we could do some comparison between primary and secondary dengue, and they didn't show major differences when you separate between the clinical presentation. And all our patients were dengue 1. We had 4 patients that were dengue 4, which were like, let's just exclude them. And so we just worked with the dengue 1 infection. Okay, thank you.
Hi, Elton. It's nice data.
Thank you.
And I'd like to ask you if you have if you evaluate samples for patients that have different genotypes of dengue 2 genotype 3, and dengue 2 genotype 2, the cosmopolitan genotype. Because Cidi cosmopolitan genotype arrived in Peru in 2019. It's the other genotype, the 3, is disappear, and you have more severe cases for dengue in Peru. In Brazil, our was detected for our group in 2021, and is increasing more than the other genotype that was circulating before.
Yeah, well, when we did our cohort, there was only type 1 circulating here in Belo. And there was 1 year that the dengue 4 started to enter in Brazil, so I was like, yay, let's get different genotype to compare, but we just got 4 cases of the dengue 4, so that's impossible to do any statistics with that. So, but here we don't have many of dengue 2 yet. I don't know, as it spreads, maybe we'll be able to do this comparison.
Okay, thank you very much, Elton. We have to move on. Our next speaker is Igor Almeida from the University of El Paso in Texas, and also now a visiting professor here at UFMG.
Not yet. I didn't say anything. So I'd like to thank Ricardo for the invitation to be here. It's always a pleasure to be in Belo Horizonte. I'm going to talk about the preliminary results we have on the Teseu clinical trial. It's a phase 2 clinical trial that we are carrying out in Bolivia. Just to briefly remind you about Chagas disease, Rafaela did an excellent introduction, and I'm not going over in detail. But I just want to remind you that we still have 6 to 7 million people infected throughout Latin America, from Mexico down to Argentina and Chile, and the disease is spreading up north. I think probably because of now the insect can be found in very high northern areas of the U.S., like they recently found in some place, I think New Hampshire or something like that, because of the climate change. We have estimated 300,000 people infected in the U.S., but this is a really a very low estimate. There's not been any survey in the U.S. So far, and we believe that this number is just in Texas, based on the data we have in El Paso. In Spain, there are about 100,000 people infected, and most of them came from Bolivia during the 80s and 90s, huge migration to Spain. So Chagas disease has been globalized. You can find Chagas all over the world, but in Africa, in Asia, but you can find in Japan, Asia, so Australia, and basically everywhere in Europe, and so the numbers are going up. So there is no clinical vaccine. There are many experimental excellent candidates like the transalidase ASP2 that Mauricio Rodrigues developed, and Ricardo and Santuza carry on the work, and probably going to be in clinical trial, hopefully, in the coming years. Not even a phase 1 clinical trial, and I hope that the CT vaccine will do that soon. In case of chemotherapy, we still have, like Rafaela said, we still have only 2 drugs, benznidazole and ifutimox. These drugs were developed during the 60s, 70s, and since then there's not a single new drug. All the new drugs have been very promising drugs have been tested. They are not very effective. So we still have these 2 drugs and have to deal with that for a while. All newer promising drugs have failed in clinical trials. In the acute phase, these drugs are excellent. They have high efficacy, 90 or more higher percent of cure, very low treatment failure. The problem is that it's very difficult to diagnose the patient during the acute phase. It is short, maybe a month, and most of the patients excuse me, are located in rural areas, like in the outbreaks in the Amazon. It's very difficult for them to find, to access public health, and by the time they arrive in a city and has a hospital or a doctor, it's too late. It's already in the chronic stage. In the chronic phase, depending how you follow up the patient, we have, and other groups have data that showing that these drugs can be as high as 80% effective if you follow up with, let's say, qPCR. The problem is that most of the qPCR data is just for 1 year, and there are many patients that after 1 year, the parasite comes back, and that's a problem. But despite the efficacy being medium-high, depending how you follow up, less than 1% of patients are being treated, chronic patients. So you still have millions and millions of people with no treatment. As to the severe adverse events or side effects. It happens in 20-30% of the patients, and that leads to permanent treatment interruption. Despite these, benzodiazole, both drugs have been approved by FDA lately. First, benzodiazole for kids between 2 and 12 years of age, and nifurtimox for newborns was and up to 18 years of age. One of the problems, some major clinical challenge we have is that the treatment follow-up or monitoring. By the conventional serology, the treatment, it takes 10 to 20 years for the patient to seroconvert, and I'm going to explain why. By PCR, PCR is criterion of treatment failure, not cure. So if there is a failure after the treatment, let's say 6 months, one year, and you can say, okay, the treatment did not work. So the patient, if he was treated with benzodiazole, you have to treat it with nifurtimox and vice versa. So this is absolutely unacceptable for the clinical settings, and so clinical biomarkers for the early evolution of therapeutic outcomes are much needed. However, there are none in clinical use so far. There are many promising biomarkers that have been developed throughout the years, in the last 10, 20 years. I'll say they're very good candidates, but none of these biomarkers have been tested in a long-term follow-up in a clinical trial. So one question we had is that why does it take so long for the seroconversion post-chemotherapy? And that, for us at least, it is very clear that like T-Cruzi and other parasites, Leishmania, other parasites, they induce polyclonal activation. So the existing B-cell clones against many different things, against fungi, parasite, and things we eat or get exposed to, they are activated, proliferate, and the plasma cells secrete low-affinity antibodies, and many of these antibodies are autoimmune antibodies. So they are found in very low titers in the serum of patients. So if you follow up the chemotherapy with the conventional serology, which is an absurd, because the epimastigote they use for preparing these antigens, they don't have the major virulence factors. Sometimes they have the virulence factors, but they don't have the epitopes involved in the virulence. So, it's the most inadequate antigen to use for the follow-up of chemotherapy. So the patients don't see epimastigotes, never will see the epimastigote. So you should use at least if you use a lysate from triple mastigote or a mastigote stage to follow up with chemotherapy. So our goal is to find biomarkers that can detect the response, either cure, or at least the patient has been responding to the treatment. You cannot wait 10, 20 years for that. So that's our goal. And so the conventional serology uses crude epimastigote lysate containing hundreds of thousands of specificities, including those that trigger low-affinity antibodies. And those who do conventional serology in the lab, they know that the dilution they use is like 1 to 1. You still use 1 to 20, 1 to 100. At those dilutions, there are a lot of polyclonal, antibodies, low-affinity antibodies that cross-react, with several diseases. So another problem is the adverse event. Can we do something about? And in this great review by Karen Byrne, she showed that the major adverse events in nifurtimox, for instance, anorexia, weight loss in up to 75% of the patients, nausea, up to 50% of the patients, headache, abnormal discomfort, and insomnia. The same thing, very similar adverse events we find patients treated with benzodiazole, like allergic dermatitis, paresthesia, and peripheral neuropathy, anorexia as well. So in a pharmacokinetic study done in 2015, benzonidazole and ifutmig have been used for over probably 70 years, and only 2015 that was the first pharmacokinetic study done in benzonidazole, and the one in ifutmig was only done a couple of years ago. So they found that the patients are probably being overdosed. They are receiving a dose which is 2.5 milligrams per kilo per 12 hours, twice that a day. And that is probably leading data from simulations in this study propose that the patients are being overexposure or overexposed or overdosed. So also dosing simulations show that benzonidazole at half of the dose and half of the frequency will keep benzonidazole within the recommended target range in most patients. And so long-term objectives are enhance drug safety and adherence and optimize efficacy of benzonidazole and ifutmig, strengthen treatment monitoring and follow-up processes to shift the perception of physicians and public health authorities regarding the effectiveness of benzonidazole and ifutmig. Nowadays, most physicians here, at least in Brazil, they don't believe that benzonidazole works because they don't have any tool to follow up, and they know that have to wait too long, for that. So we proposed this study called Teseu study. We see here a statue of Theseus and the Minotaur, and for us, the Minotaur is the Trypanosoma cruzi. Hopefully, we'll be able to, somehow kill this Minotaur, or at least, attenuate this disease for most patients that can be treated. So this is a clinical trial we started in 2018, just 2019, just a few months before the pandemic, and was very bad luck, and I'm going to tell you more about it. So the goal one of this study is to test new dosing regimens for benzonidazole and ifutmig. So based on that PK data, so we propose that half dosing frequency could maintain therapeutic levels and reduce adverse events while keeping at least keeping the same levels of drug efficacy. And assess the efficacy and safety with 30-day treatment at half benzonidazole dosing frequency to maintain drug efficacy while improving safety, and also explore efficacy with 90-day treatment duration and reduce dose frequency to enhance treatment efficacy. Because, the 60 days treatment was based on treatment of animals, of mice. We never did let's say, a non-human primate study with benzonidazole and ifutmig. Went straight from rats and mice to humans, and that it doesn't work. So in most cases, it worked in the case of benzonidazole, say. And goal two is to evaluate novel potential biomarkers in response to a specific treatment, eventual parasitological cure in chronic Chagas disease. So a biomarker is a molecule either from the host or from the parasite that can be detected, by molecular methods or serological methods that we can use as a marker indicator of cure or at least response to the treatment. So we propose two classes of biomarkers based on host-derived biomarkers, meaning antibodies. The first one is the anti-alpha-gal antibodies. We're going to use purified TGPI mucin. There have been several people have talked about these in the last 3 days, Ricardo and many other people, and synthetic alpha-gal and beta-galactofuranols as well, which is another sugar that we don't make it at all, containing new glycoproteins. And also going to use recombinant proteins from HSP70, paraflagell ROD2, and PEP3973 from a group in Granada that has very interesting, very promising preliminary data on that. And finally, you're going to use parasite-derived biomarkers, try to detect them using focus on these triple mass secreted antigens, or TESA, that was developed many years ago by Umezawa in São Paulo. And Alain de Brabant lab at FDA use TESA-specific aptamers to catch, these secreted antigens. Mainly, most of them would be like mucins and transalidase and MASPs, which are secreted via extracellular vesicles. Our hypothesis is that by having the dose and frequency of benzodiazole and nifurtimox, their plasma levels should stay within the therapeutic range, and to the effectiveness of benzodiazole or nifurtimox may be linked to treatment duration, so proposed to a longer treatment, and blood levels of proposed biomarkers should decrease rapidly following treatment in contrast to conventional serology. So our trial was carried out in Bolivia. It was a phase 2B open-label, randomized, prospective, observational study with 450 patients, participants in 6 parallel arms, 75 per arm, no placebo arm because we consider that complete unethical at this point do a placebo when you have so much data on placebos in the same region. Have at least 3 or 4 clinical studies done in these 3 sites. So these 3 sites are just pre-Andes. They have the same geographic traits, altitude, climate, and demographics. And why Bolivia? Because Bolivia still has the highest Chagas disease infection rate in the world, about 10-12% of the urban population, and 40 to 60% of the rural population. So in previous, also because we carry out several clinical studies, I participate in these studies, phase 2 clinical trials performed exactly at the same site with the same population. So no reason to use placebo. So we're going to have 6 groups, 6 arms, and the inclusion criteria is that adult patients, 18 to 50 years of age, infected with T. cruzi with 2 positive conventional serology tests and at least 1 positive qPCR, and indeterminate form, no clinical manifestation, or early cardiac stage, Kushner stage 1. So we have 3 arms with benzodiazole. 1 is standard of care, 150 milligram twice a day, 60 days. The other one is half of the period and half of the dose, just once a day, 150 milligrams. And the third one, 150 milligrams once a day for 90 days. In nifurtimox, we have the standard of care, which is 60 days, 240 milligrams. And nifurtimox for 30 days. We could not use half of the dose because there was no evidence, not even experimental evidence that suggested that it could work. So we only proposed things that we had some evidence in the literature, either experimental but better derived from clinical trials. So, and the last one is 240 milligrams once a day, 90 days, because there was a data from an assay that group of in Argentina carried out a long time ago. So that's the trial design. I'm not going to enter in detail because of the time restrictions. So we treat all the patients already. They're done. We're done with the follow-up. We have 36 months of follow-up. That's the first trial ever to do a 3-year follow-up. And was very difficult because we are hit with COVID. So we start the trial in December 18, 2019. And in March, the recruitment was installed. And the rate that was going very fast, we're probably going to end the recruitment like 6 months earlier. We only ended much later because people are very afraid to go to the sites, to the study sites. However, we lost no patients during the 3-year follow-up. In fact, this is, and for that we designed an algorithm. I'm not going to enter detail, but to use IgM and IgG to see if the patient was in the acute stage or chronic stage of COVID, and then we could recruit them. So this is the data, time to first adverse event, drug-related, so we call dry. This is for benzodiazole, and you can see that benzodiazole 30 days arm, have much less cumulative adverse events in comparison to the standard of care 60 days and the 90-day. When you look at the nifurtimox, we have many more adverse events, drug-related, And perhaps the nifurtimox 90 days had much lower cumulative number of adverse events. So when we compared in terms, this is the resolution, severity, and treatment action related to the DRIES, we find that we see the benznidazole, just considering all the groups, you see much less adverse events in terms of severity. You can see here, mild 141 versus 450, moderate 120 versus 304. So basically everything is, you had a huge p-value, and also for those adverse events that led to permanent interruption, we can see that they had about a little bit more, 50% less than the nifurtimox treatment. This is distribution of drug-related adverse event. You can see that we have many more adverse events that presence of DRIES in nifurtimox group in comparison to the benznidazole. So the take-home message that follow-up concluded on May 29, 2024, and 36-month follow-up adherence rate of 97% we had, and also the adherence to the treatment was 86%, which is pretty good. Overall, 91% had negative qPCR. Overall, we don't know the groups because it's still blind for us. And short and longer benznidazole regimen, 90 days, are very promising, but we don't have the qPCR data. Can I have 5 minutes to finish? Yeah. So currently performing immunoassays with these biomarkers, and the qPCR data will be unblinded by probably August or September this year. So in terms of biomarkers, so we have been studying the surface of the parasite. I think translational research without basic research doesn't exist. You just, trying, to propose things based on a black box. So we have to know the molecular level, what's going on with parasites. So I have been studying this parasite surface since the 90s when I started my PhD. And we know that mucins and transalidase and MASP-associated surface proteins are major virulence factors and surface glycoproteins. In the mucins, you know that they express alpha-galactose, which is an epitope that's extremely immunogenic to humans, and you don't have this linear alpha-gal epitope. Whenever the patient sees this epitope in the parasite, which is immune dominant, they make a very significant amount of lytic antibody that can bind, agglutinate the parasite and kill, and correlate with the active infection. We showed first time in a study published in 1996 in Lancet. That was the first evidence of any biomarker, serological biomarker, where you saw that after 3 years, kids treated with benznidazole, 58% of them, they crossed the cutoff for the anti-alpha-gal antibodies in comparison of 5% in the same time, and after 3 years more, 85% of the kids were negative for the anti-alpha-gal antibody. So how we can replace TGPI mucins, which are not feasible for large-scale clinical application due to purification complex and batch variability with surrogate synthetic alpha-gal glycoconjugates. We synthesize a series of biomarkers. We did what we call reverse immunoglycomics. Instead of taking the parasite and characterizing all the glycoconjugates, we did the opposite. We start from the bottom up. So we synthesize first and then look for the antibodies that were found in serum of the patient. We synthesize a library. In total, we have about 40 alpha-gal compounds. And then we can couple them to a carrier protein because sugars alone, they cannot bind, stick to any hydrophobic surface. And then you check by MALDI before conjugation, alpha conjugation. We can determine the number of unities that are attached to derivatized succinimide derivatized BSA. So and you see when you screen with serum of patient, you can see that the linear trisaccharide, which is the immune-dominant epitope, is the most important. But if you remove the glucanac and put glucose, it's the same thing, meaning that most of these anti-alpha-gal antibodies, anti-glycoconjugate antibodies, they usually recognize just the disaccharide. So anything after that, it doesn't matter, it seems. You can see here the disaccharide is as good as the trisaccharide. So we did preliminary studies in Bedita trial, which is a trial that was published years ago. And we can see that both this NGP24, which is the trisaccharide, can see overall drop in geometric mean titers. And when you look at the mucins, also you see the drop. You can see there's a p-value, very significant p-value. But this doesn't mean anything. You have to look at each individual patient to see the kinetics. And when you look at the kinetics, we see two groups. 60% of patients respond to the treatment, and 40% did not respond to the treatment. And you can see here very easily in these two graphs, representative graphs, that this patient will probably going to be able to predict when the patient is going to cross this cutoff. And this is a patient that did not respond to the treatment. So here, as early as 6 months, we'll be able to say this patient did not respond to the treatment. We have to treat with the other drug. So, and then we're going to do the same for the Teseo clinical trial. One problem we had is that we have 10 selected biomarkers, and we have 450 patients, not sample patients, and that means about, we have 10 points, time points for each. That's about 4,500 points, and that we're going to take like 450 plates, ELISA plates, and 10 biomarkers. That means we have to run 4,500 plates, which is a pretty bad task. So we developed this glycoamine array called GIA. We bought this nanoplotter, and we plot these glycoconjugates in this ELISA plate in each well. And these are the 10. There are different versions of the alpha-gal epitope, alpha-1,2, alpha-1,3, alpha-1,6, and also beta-galactofuranose epitope, which is very, very immunogenic. And this is a control epitope that doesn't have alpha-gal or beta-galactofuranose. I'm just finished here. So this is the array. This is one ELISA plate, one ELISA plate per patient. And we can see now each one of these ELISA, we're going to give 20,000 data points for each patient. So this is the reactivity of Chagas patient, and this is the control of normal hemisphere. You can see there's no reactivity in the middle where the new glycoproteins are. This is our reference spots, and these are calibration curves. And here we have the different new glycoproteins in different concentrations. That's the result we get, the heat map. That's the control, the negative control. And this is negative sera from Bolivia, and this is positive sera from Bolivia. You can see the difference, a huge difference that goes up to 20,000 ratio, signal to noise ratio in comparison to the negative samples. So we validate using the Bedita trial sera. That's the same thing, heat map. But the most impressive thing that we see that most patients treated with standard of care, you can see the curve. We can see this patient is going down. Some of them are already seroconverted for some of the biomarkers. And that's another group treated with 4 weeks. Half of the time, you see the same, even better profile, depending on the patient. So you can see can do early assessment of treatment. This is a patient treated with E1224. Intermittent treatment, you see this patient did not respond to the treatment. And you can see this after 6 months, the reactivation of the PCR. And here of the serological titers using beta-galactofuranose. So this is placebo group. It doesn't respond to the treatment. You have this variation of parastemia in the patient. So take-home message, there's synthetic alpha-gal new glycoproteins, beta-galactofuranose new glycoproteins, akin to GPI mucins from triple mass goat strongly and specifically react with chronic Chagas disease serum. These new glycoproteins are surrogate biomarkers of these mucins for AL assessment as early as 6 months in comparison to 10 years, 20 years by GIA. or therapeutic outcomes. We have clinical trials that we finished, analyzed. The Bendita trial just finished the analysis. Facts we're going to do next, but this is done. This trial is over. And we're going the ongoing biomarker immunoassays for the Teseu 3-year follow-up. So I'd like to thank it's a huge group of people involved, and thank especially the NIH for the funding for this study. Thank you, and I'll be open for questions.
Thank you, Igor. I think we need to give a message here, Ricardo. If you have pressing questions, please come to Igor afterwards, but Ricardo has a...
Fantastic work, Igor. It's really... but because we are really on...
no, it's okay.
We're going to leave people come to you and ask questions. Just to... So we come back at 1:15. It's very important because our diamond sponsor, which is Thermo, they're going to start their presentation, and I really want to have people here to watch their presentation. So if you can help with that. Thanks a lot, and see you